We can kick off. I think everyone now is out of the waiting room. Everyone is in the call.
Okay. So hi everyone. Today is in our August webinar. It is our great honor and support that we are welcoming two renowned scientists who will share their insights about the scientific journeys regarding climate change impact on the plant adaptations that farmers outside Europe are also facing this challenge, as well as the African subsistence crops. And also, they will give us some important advice regarding what matters in building competent scientific career, especially for students and unexperienced researchers like us. So please briefly let me introduce our guest speakers. So I would start with the lady first. So the Dr. Patient Chatukuta, she's a research group leader at the Max Planck Institute for Biology Tübingen in Germany. Her research group employs the genome-centered approach in order to study the diversity of under-researched subsistence crops in Southern Africa, together with several scientists based in African continents. Her research group also focuses on understanding horn melon diversity, crop improvement through DNA-free genome editing, and African biodiversity conservation. And the second speaker is Professor Dr. Detlef Weigel. He is an adjunct professor at the University of Tübingen and a director at the Max Planck Institute for Biology in Tübingen. Professor Weigel's research group had led a grand collaborative work in establishing the Arabidopsis 1001 Genome Project that shapes our understanding in genetic variation in plants. And his lab currently focuses on plant evolution and adaptation, plant microbe interactions, and also plant immunity. Professor Weigel has been contributing significantly not only scientific discoveries, but also being a great supportive mentor, academic father, and a role model leader who has been supporting scientific outreach and education in plant science. So he has been also one of my great figures that I am learning from based on my education development in Germany. So please welcome our speakers. And I think Professor Weigel would lead the story first. Thank you so much.
I thank you so much for the invitation, and thank you for to everyone who has time to join us today. As Prachaya said, I'm a director at the Max Planck Institute for Biology. Dr. Patience Chatukuta is a group leader at the same institute, and I'll start off. I tell you a little bit about the more fundamental research that's going on at the institute in my own lab, how we've been thinking about genetic variation, and then Patience will continue and speak about her work looking at genetic and phenotypic variation in crop plants. I will share my slides right here and start right away. So, all right, good. Mm-hmm.
We can see it.
All right, so I think no one needs to be reminded that there is global change. And when you look at how the climate is changing, actually one of the things that is going to be the greatest threat is going to be more extreme droughts. And this is from a paper that's already quite a few years ago, was published quite a few years ago, looking at a measure of a potential water deficit called Permadrought Severity Index. And of course, you see prominent over here, Southern Africa might not be surprised there. You also see the Amazon is very much at threat. But some of the most extreme threat, this might be, I think, surprising, was certainly surprising to me when I saw this, is here in Europe. And so you might be asking yourself, why is this? Because Europe is often considered to be quite a rainy place. But because it is relatively rainy, there's also the greatest potential for a change. So for things to become much more drought prone. So if you look here, for example, in the Sahara, there isn't going to be much of a change because it's already really dry. If anything, it might become wetter in the Sahara. Now, plant scientists have been thinking about how wild plants adapt to climate change for many, many years. And the conventional scenario is that plants adapt just by migrating. So, for example, imagine a plant that has a more southern distribution in Europe, or a plant that is in lower altitudes. Prediction is that these plants will either move northwards or will move up the mountain. And when you look at papers that have been published, typically they have these sort of predictions here. This from a paper that's, again, already quite old, over 20 years ago, looking at a particular species distribution today, really 25 years ago, then what it would like in 2050 was moderate climate change, or 2050 was extreme climate change, and you see this northward shift. Now, this has a bit of a problem, because these sort of scenarios actually rely on the least adapted, the least diverse populations to move. So this is just fundamental effect of populations, that in the center of any population, you're going to have the most diversity and the most potential to adapt. And then here at the margins, you actually have small, fragmented population, limited genetic diversity. So these are actually the least prepared populations, if you will, for adapting to new climates. Yet if you have this move, obviously these populations would play a very important role. So what we wanted to ask is, what is the potential of plants when they don't move, when plants are in the place where they grow now? What is their potential to actually adapt to a changing climate? And we've been doing it with this plant, Arabidopsis sayana, that many of you might have heard of. It's a small, weedy plant that has its origin actually in Africa, is today most widely distributed in Europe, into Asia, with some populations also in Eastern Asia, and then it has been introduced to North America, to South America, and also to Australia and New Zealand. And so when you look at where this plant grows, you see it in many different habitats. So here, for example, on a railway, abandoned railway. Here is an oak forest in Spain. Here it is growing actually on a beach, the Baltic Sea. And here in Patagonia, at the southern tip of South America. So just looking at this, you see that the plant can grow in many different places. And of course, also the geographic distribution. You see it around the Mediterranean, you see it near the Arctic Circle, you see it at the Atlantic, you see it in Central Asia, and so on and so forth. So you know that this must be a pretty adaptable plant. So the experiment that Moy Exposito Alonso, who was a graduate student at the Insitute, then became a group leader at the Carnegie Institution, and now is actually a faculty member at UC Berkeley. Experiment that Moy set up as a graduate student was a pretty simple experiment. Take accessions of Arabidopsis sayana from throughout its range, and then grow it in two different places. In Tuebingen, where naturally precipitation, rainfall is relatively high, and in Madrid, where normally rainfall is relatively low. And grow it in these tunnels here, so one could control actually the amount of water that these plants get. And so you won't be too surprised. You look at the plants here, so these are many different sections, so many different varieties being grown in a certain experimental design. And then these plants here are well watered, and these plants here are ones that are exposed to mild drought. And you see right away that these plants do much better than these plants over here. And so we did it in these foil tunnels to otherwise have natural temperature and natural humidity, and also natural pests and all of this. So in the end, we had 24,000 pots. They were photographed at 20 time points each, and then we counted how many seeds survived and how many seeds actually these plants produce. And so this seed counts gives you a measure of the fitness of the plant, because it's a definition of Darwinian fitness, how many progeny an animal or a plant produces. And we did that both for individuals and populations. So you see here, we had some pots where we had just a single plant, and then we had other pots where we had 20 plants together. And because having neighbors or not having neighbors also influences your survival. So looking at this, you can see here in low density and high density that you won't be surprised. The plants that had rain conditions, they were much more likely to survive. This just gives the number of seeds that they produce. The plants that were under rain conditions, they are much more likely to survive and produce seeds than the ones that are under drought conditions. And then in Tuebingen, in generally, it's not as hot and the air is not as dry, so plants do better in Tuebingen than do in Madrid. And you also see here high density versus low density. Plants produce many more seeds when you grow them at high density compared to low density. So here you have a single plant, and here again you have these 20 plants, as I mentioned before. So what can we do with these data to learn about adaptation? Well, what we can do is we can go along the genome, and along the genome, we can ask for every gene or every genetic variant, we can ask whether a variant in that place in the genome increases fitness, the number of seeds, or whether it decreases fitness. And this relies on the roots of a long-standing project that Tachaya already mentioned, the 1001 Genomes Project for Arabidopsis Genomes. So all these strains that were used, about 500 different strains, they all had completely sequenced genomes, so that we have millions and millions of SNPs that we could interrogate. So it's really super simple. We go along the genome, we ask, is there variation? If there's variation, we classify all the strains in having either one allele or the other allele, and then we just calculate how many seeds on average are made if you have this one allele, how many seeds on average are made if you have this other allele, and we just get a difference and can plot that along the genome. That gives you a map of alleles that are either favorable or not favorable for survival under these conditions. But then the interesting thing is we know where these accessions come from. So we can ask for every allele, where does it come from? Now, of course, the accessions, these strains that have the different alleles, they are distributed over a geographic range. So there's not any one place, but we can draw a circumference, and then we can ask where is the centroid of this distribution. So if we do this, we can ask alleles that either support survival in Madrid or that are acting against survival in Madrid, where do they tend to come from? And we split this up here. What is the annual precipitation at the place of origin of the different alleles, and also the precipitation seasonality? And that means, does it rain evenly throughout the year, or does it rain not so evenly throughout the year? So in Tuebingen, it rains more or less year round, so precipitation seasonality is low. In Madrid, it tends to rain not a lot in summer and much more in winter, so precipitation seasonality is higher. And then the amount of precipitation is lower in Madrid than it is in Tuebingen. So what you see looking at alleles that are favorable in Madrid, you see they are much more likely to come from a climate that is similar to the one in Madrid, so with low annual precipitation and relatively high precipitation seasonality, whereas alleles that act against survival in Madrid, they are much more likely to come from a climate that is similar to the one in Tuebingen, with high annual precipitation and low precipitation seasonality. So this tells you that home alleles on average are much better, which is sort of expected from what ecologists have done for many, many years, where they took plants and transplanted them from one place to another and asked, do the plants do better in the place where they come from versus the plants you transplanted them to? So that's sort of expected, but it hadn't really been shown at the genetic level, that you can really, show this for individual genetic variants and not just for the entire genome. So that, of course, then begs the question, if plants are adapted to the local climate, can they adapt to a changing climate from the variation that is present in a place? Now, in a specific place, of course, you have many different strains. The different strains carry different alleles, and you can imagine that the strains cross. And if everyone has a few alleles that are favorable in the drought, for example, you could cross these strains together, and what could emerge are lines that have many of these drought favorable alleles, and then actually in situ, in place, new genotypes could emerge that are actually then able to deal with the changing climate. So Moy did another experiment to ask this directly in an experimental setting, where he took, again, hundreds of accessions from the 1001 Genomes Project, and he did very simple experiments, just asked whether plants are able to survive extreme drought. Now, a small weedy plant like Arabidopsis Genomes, many weeks without rain, that is typically what kills them. So this is what it looks like. So initially plants were watered, and then water was withheld, and water content in the soil dropped to a very low amount. You can also see here there was a little accident. There was one of the student helpers hadn't gotten the memo not to water the plant, thought the plants looked really sad, and then re-watered the plants, and that's why all of a sudden soil water went up where it should not have been. So this is what it looks like then. This is images of plants over time. And you see the plants disappear. It's actually not the plants disappear, but we use here actually only the green pixels. So the green pixels disappear. There are some plants which become yellow and die, and that's why these plants disappear quicker than other plants. We can then estimate from the images the survival by just counting the green pixels for each of the plant over time. You get these curves here, as you see here. So first the plants grow and they are watered, so the number of green pixels goes up. And then as we withhold the water, pixels go down, and you can see some of them, green pixels disappear much more quickly than for others. So we can calculate a very simple drought survival index by approximating this one here with a quadratic equation, and then looking at what is the quadratic parameter. And so see here different quadratic parameter for the plants that survive quite long and have managed to stay green versus the ones here that do not survive as long, and by the end of the experiment are dead. We then can project this information back on the map. We know where every one of these strains comes from. We also know what the climate is like in the different places where these plants come from, and project that back here on the map and ask where are plants likely to survive drought. You see here, as expected, around the Mediterranean, plants are expected to survive drought quite well. Plants here in Central Europe and in the north of England and Scotland and Ireland are not expected to survive drought that well. The east of England does better. That's not so surprising because the east of England is actually quite dry. It's drier than most places on the continent. And then a little bit surprising is here that in Sweden, for example, plants are predicted to be quite drought tolerant, which at first seems somewhat counterintuitive. But the explanation is actually these plants here spend a lot of time under snow. And under snow, as you actually have drought conditions because all the water is in the snow. So you can take this information and then again make a map of where you have good and bad alleles for survival under drought. So this is just a markup. So in yellow are the bad alleles, and purple are the good alleles. And you can have then an estimate of the density of good and bad alleles at every point on the map. You see here again, so in the center of Europe, you have very relatively few good alleles supporting drought. And then here around the Mediterranean, and obviously here in the north, you have more good alleles supporting drought. So this is what it looks like today. We can ask in future climate, how many of the alleles do you need? How many more alleles do you need as they are today? So this would be the number of alleles that you need, additional alleles that you need for survival versus the alleles that you need today. And you can then calculate an adaptation deficit. So the number of alleles that you might need to survive and how many alleles are actually present in a specific place on the map. And so what you see here, this is an enlargement, here in Central Europe is actually the greatest adaptation deficit. So these populations here, they have some alleles that are helpful for survival of drought, but not nearly enough for what would be needed to survive a future climate. So what we learn from this is that when we ask who is likely to survive in a future climate, it's actually not the plants here around the Mediterranean. They are already well adapted to drought because there you have a lot of drought today. But it's actually these, the vulnerable populations are the ones here in the center of Europe because they haven't experienced drought historically. So we see that some populations can actually adapt to a changing climate, others cannot, just based on the variation that is already there. So the final question that we wanted to ask is, are new mutations potentially limiting for adaptation? So for a long time then, what my lab has been looking at is what is the rate and spectrum of new mutation. So there are many sources of mutations. Some are endogenous to the plant, such as replication errors, activation of transposons, and then other sources are external to the plant, such as sunlight or toxin. And so most DNA damage is, of course, repaired by several different mechanisms, but of course, this is not perfect. And when something is not repaired, that is when you get a mutation. And this is something that biologists have thought about for a long time. What is the natural mutation spectrum and rate? It's, of course, absolutely essential for understanding evolution. And the way this is typically done is to generate these what's called mutation accumulation lines, or MA lines. You take an ancestral line and you just propagate it for many generations. And then at the end, you ask how many new mutations are there. Now, this, of course, takes a long time. And before sequencing of genomes was possible, Muller, who received the Nobel Prize for discovering mutation, he actually thought that mutation is something that we all know must exist, but it might never be observable. It turns out it is actually observable, and this is one of the first applications where we use short-read sequencing. We used lines that had been maintained for something like 30 generations. And then after 30 generations, we sequenced their genomes, and mutations are rare. Each of these plants had only a handful of mutations in their genome, but enough that we could estimate the mutation rate. Works out to about 1 mutation per haplogenome in generation. That's for Arabidopsis genomes, which has a relatively small genome, and we expect this to scale. So if you have a genome that's 10 times as large, the mutation rate per base pair doesn't change, but the number of mutations will be about 10 times more. And that's about 0.2 non-synonymous mutations per haplogenome in generation. That also fits quite well with what had been estimated from how fertility and fitness declines over in these sort of experiments. So this was done in the lab. We, of course, wanted to know whether this also holds true in nature. By luck, really, we discovered what looked like a natural mutation accumulation experiment. When we had looked at North American Arabidopsis thaliana, we saw many strains distributed all over North America, or the purple ones here that seem to be almost genetically identical. And so if this is a natural mutation accumulation experiment, it would have been something like this, that Europeans, when they colonized North America, that inadvertently they brought seeds to North America, and they, in the historical time, then acquired mutations. And so in principle, one could figure out what is the mutation rate by sampling plants that have been sampled, have been collected at different plants at times. And so we have, for example, plants were sampled in 1990, and we have plants that were sampled in 2000, but 10-year difference is not a whole lot, so we don't expect too many mutations to differ. So it would be really great if one could actually, sample some of these plants that from 100 or 200 years ago. Now, and then we would have bigger difference in the number of mutations here versus the number of mutations here. Now, for doing this, of course, it would be great if we had something like a time machine. We all know that doesn't exist, but there's something really similar to a time machine, and that is herbarium. Maybe Patience is going to say something about the wonderful work she is doing with herbaria later. So there are herbaria where there are plants that have been sampled in the past, and one has to use certain precaution to sequence the DNA to make sure it's not contaminated with modern DNA. But when you do this, you can actually determine what is the mutation in the plants that have been sampled in the past. So we acquired such a data set where we had plants that had been sampled from 1860 all the way to the 2000s. And when we do this, we can, again, estimate the mutation rate. And what we find is that the mutation rate is actually very similar to what we had measured in the lab. So what does that tell us? Well, what it tells us is that there is this vast universe of mutant plants. So here you're looking at the wheat field, and might look at this and say, well, these plants are all identical because they all come from the same strain. But actually, back of the envelope calculation tells you that each plant has about 200 mutations in its genome. And so if you extend this further, if you look at a field of wheat of just 1 hectare, you actually have something like 200 million mutations here. So that the entire genome is basically mutagenized here. Sorry, 200 million grains, and you have something like 40 billion mutations, sorry. So 40 billion mutations in the harvest from a single hectare, where basically the entire genome has been mutagenized. So the conclusion here is that actually mutations will not be limiting for adaptation. There are many other processes that are, of course. So this is not an excuse not to work hard to prevent climate change, but when plants can adapt, it's not because the mutations are limiting, but because of other forces. And that's already all I wanted to say. So as I think you saw, we are doing basic research trying to understand evolution of plants. And it's really through this kind of research that I had started to think about genetic diversity in crops. And this then led to Patience coming to the Institute and setting up her own research program on genetic and phenotypic diversity in plants that are a little bit more important than Arabidopsis is. So I'll turn it over to Patience, and I suggest we wait until the very end to take questions both together, Patience and I. So Patience, the floor is yours.
Yeah, while Dr. Patience is sharing the slides, just in case you have questions, but you worried that you may forget about it, you can type into the chat as well, and we'll come to that at the end. We can see the screen now, the Patience.
Yeah. Okay. Thank you, Detlef, for the nice introduction, and Pachaya as well. Thank you to the team.
Quickly, sorry, I think we're seeing the presenter mode.
Oh,
maybe.
Sorry. Let me.
Just good.
Okay. Is that better?
Yep.
Yes, perfect now.
Okay. All right. Good. Perfect. So thank you, Detlef, for the nice introduction. And thank you, Pachaya, and the team at SynBio4ALL for inviting me to this webinar. And so Detlef has given a really nice introduction to his study of genetic diversity in model plants, and we'll be shifting to subsistence crops. And so one of the things that really sort of attracted me to the Weigel lab was the scale at which plant genomics data was being generated. And coming from South Africa, where I was living at the time, it was mind-blowing that this lab was actually sequencing the genomes of hundreds of accessions of a single species in order to answer questions around genetic diversity. And that was around 2021 when I was reading this paper that had already been published in 2016. And it was showing that they sequenced genomes of accessions from Eurasia, North Africa, and North America. And so I scanned the literature to find out what was happening with respect to plant genomics in my neck of the woods in Africa, because at that time, single sequencing of even a PCR amplicon was a really big deal where I was working in South Africa. So here's what I found. Africa holds 25% of global plant diversity. And by 2021, only 20 African plants had been sequenced out of 798 plants globally. And none of these plants had been sequenced on the continent. Actually, they have been sequenced in China, in Europe, and in the United States. And so the participation of Africa in this fast advancing field of plant genomics was minimal. And African scientists were actually taking more of the fieldwork roles, and they were not really involved in the data generation or analysis roles. And we know that plant genomes facilitate genetic diversity studies, functional genomics, comparative genomics, smart breeding, plant conservation. But the problem was that in the absence of genome sequencing of African crops that is driven by Africans and serves African agendas, the prospect that the use of African genomes would be used to drive research and development elsewhere was all too real. At the same time, scientists in high-income countries were celebrating the fall of the cost of genome sequencing and the emergence of new long-read technologies. But the cost of this tech was still too high. It was still out of reach for African scientists. And so it was starting to seem as if plant genome sequencing on the African continent is impossible. And the question then became, how best can Africans leverage partnerships that are mutually beneficial with high-income countries, where the agenda of the African partners plays an equal role, and where the African partners have a say in the species to be sequenced and how the data is used. And since then, since 2021, a few plant genomes have been sequenced locally in Africa, including yam bean, lab, K-apple, and the king protea. And what is remarkable for me about these papers is that the projects were all African-led international collaborations. And I hope that you noticed that 3 of these genomes that are reported here are actually subsistence crop genomes. And the lesson for me was that given a chance, African scientists will prioritize research on subsistence crops that are locally adapted, that are climate resilient, and that have potential to enter large-scale agricultural production. And that is exactly what we're doing in the African Plant Genomics Group. We are using these lessons of leveraging resources through international collaborations to develop genomic resources for horned melon. Horned melon is a subsistence crop of the cucumber family. It is indigenous to Africa. And we have established the horned melon as our model crop, and we are developing genetic tools to enable molecular research on this crop. So the crop itself is an annual plant. It has a short growing time. It can produce mature fruit within 2 months from germination. Its genome size is relatively small for plant genome. It's 390 megabases large, and it also has a heterozygosity of 0.3%. The species is amenable to inbreeding and outbreeding and outcrossing as well. It is closely related to cucumber and melon, but it is sexually incompatible with these close relatives. We have access to a germplasm collection that includes wild types, land races, and modern cultivars. And the crop is an important subsistence crop for smallholder farmers in Zimbabwe, where it originated. We have started working with scientists at the University of Zimbabwe to build knowledge around this crop. And we're really starting from the basics of documenting the information about the traditional knowledge around it, its geographic distribution within the country, its phenotypic diversity, and we're taking our study all the way to genome assembly of multiple accessions. And our research tries to answer questions relating to how the genomic structure of horned melon is associated with the diversity of its physical characteristics, including domestication traits, agronomic traits, and also its uses and its geographic distribution. So to begin the study, we conducted a survey of 300 farmers across Zimbabwe. These are mostly rural farmers. And we wanted to document their knowledge about the uses and importance of the crop. And we found that about 60% of these farmers are women. And therefore, it's important for us going forward that any human involvement of the project is gender sensitive, as a lot of knowledge around this crop is carried by these women. And we also found that the crop is thoroughly domesticated. So farmers primarily grow the crop in their fields, but a small proportion of people also gather the fruit from the wild. For those farmers who are cultivating the crop, we found that horned melon is a secondary crop, as they are intercropping it with commercial crops such as maize, sorghum, sugarcane, tobacco, and cotton. And when harvested, the fruit is generally eaten raw, although a small proportion of people may cook it and eat it as a cooked vegetable, and they may use it in traditional medicine or in stock feed. The crop can be cultivated in 3 growing seasons per year. Generally, it is cultivated for 1 season during the rainy season in Zimbabwe, but some of the farmers reported that they have grown it in 3 seasons during the year. And the fruit has a shelf life, a really long shelf life of up to 2 years at room temperature. We also learned an important lesson about asking questions appropriately when we were speaking with these farmers. So when we were asking them how they use the crop itself, we had given them 4 options. So they indicated to us whether they eat it raw or cooked or in traditional or they use it in traditional medicine, or they use it as stock feed. But then when we asked them in a follow-up question about what ailments can be treated with the crop, then their responses covered 12 conditions, where the farmers have observed alleviation of symptoms when treated with horned melon. So this included diabetes, hypertension, peptic ulcers, gastric acid, menstrual cramps, postnatal pain, and fontanelle closure. And so it became apparent to us that when we referred to traditional medicine in the crop use question, the farmers associated traditional medicine with potions that are used by traditional healers. And they were really not thinking of traditional medicine in the sense of how they use the crop to relieve some disease symptoms on a day-to-day basis. And so these are some of the interesting things you learn about when you talk to the people who are growing these crops. We had noted that the publicly available occurrence data for horned melon in Zimbabwe was documented mostly in the 1980s, and it was now outdated. So in order to update the occurrence of data, we surveyed government agricultural extension officers on their sightings of horned melon, and were able to fill in gaps in the publicly existing data sets. So, for example, the Mudzi district here in northeastern Zimbabwe is not represented in the publicly available data sets, but the Agri-Tex officers had observed the crop growing in many villages across Mudzi district. So the horned melon grows in these specific locations shown here, and it grows in many more locations within this district that we were not able to map because they were identified only by village names that are not available on Google Maps. And so this situation persists for the rest of the country. So we're able to fill in more occurrence gaps across the country. And with this updated map now in hand, we then proceeded to start building our own germplasm collection so that we have easier access to the germplasm. So at our institute, we obtained accessions from the gene bank collections of the USA, Germany, and the Czech Republic. These collections were made in the 1980s, and they represent these specific locations in Zimbabwe. And we needed a collection that represents the current distribution of the crop. So we then embarked on a nationwide germplasm collection exercise that is actually still in progress. And we have collected 200 accessions across Zimbabwe to date. And now that we knew where the horned melon is occurring and how it is used, and we had a more representative germplasm collection of 230 accessions, we then proceeded to characterize the morphology of the accessions in our collection. So our study of the phenotypic diversity in horned melon shows diversity in traits of agronomic importance, such as a fruit shape, coat color, spike size, pulp color, and leaf shape, and yield and weight as well. And these are just a few of the descriptors that we recorded. And so now that we know what the diversity of the phenotypes in horned melon are, we are keen to know how this phenotypic diversity is associated with genotypic diversity. And to start, we assembled 2 reference genomes from both ends of the phenotypic spectrum. So we assembled 1 genome of a wild type, and the second genome was of a modern cultivar. And we assembled these genomes using PacBio Hi-Fi long-read data. And we found that the genome sizes are around 390 megabases, and they're at least 73 megabases bigger than a previous genome assembly that was made using PacBio CLR data in 2017. So these will be our reference genomes for this project going forward. And the accessions that are being collected in Zimbabwe will be resequenced in Zimbabwe to generate Oxford nanopore long-read data, and in Germany to generate Illumina short-read data. We also found from our survey that the farmers' biggest challenge is pests, particularly Lepidoptera lavi, which damaged the fruit. And the farmers also had challenges relating to access to very few varieties. And they also were seeing more instances of extreme weather conditions, and they had limited access to markets. And so essentially, they would like to make more money from this crop. And with respect to traits, the farmers' biggest ask is that they would like a variety that is resistant to this specific Lepidoptera pest, which species we have not identified yet. They have also indicated that they would like crop improvement for bigger fruit size, a sweeter fruit, fewer seed within the fruit, better drought tolerance, and also reduced bitterness of the fruit. So the genetic data that we are generating will be used in the short term for genome-wide association studies of the important agronomic traits. And in the long term, this data will be used for genomics-assisted breeding of the crop by scientists in Zimbabwe. And I'd like to mention that this project is also a conduit for capacitation of plant genomics research at the University of Zimbabwe. So Zimbabwe gets quite a lot of its income from agricultural production. It has a very long history of crop breeding, but it has been lagging behind in terms of incorporation of genomics into crop breeding strategies. And so the university benefits through this project via training of their personnel and students, acquisition of equipment, and access to data. And I hope that this project will be one of many success stories of subsistence crop genomics in Africa. And so I didn't mention before that our group is quite young. We are about 1.5 years old. And so during the past year, we have learned a few lessons, and I would like to share some of these lessons with you. So we have found that Zimbabwe has a significant but unrecognized horned melon diversity, which plays a role in rural nutrition and farming systems. And I think if you replace Zimbabwe with Africa and horned melon with subsistence crops, the sentence would still be true. So Africa also has a significant subsistence crop diversity, which plays a role in nutrition and in farming. One of the challenges that we came across was that there was not much background data to guide our research, and these data gaps actually hinder strategic planning for the use of subsistence crops. And therefore, it is really, really important that you must systematically document all the information that you can around these subsistence crops. And this documentation can reveal the potential of locally adapted crop improvement or locally adapted traits for improvement and also for income generation. We have also learned that the local scientists have a lot of expertise, and this expertise is really, really crucial to the success of these international partnerships. So you cannot have science in Africa without involving the African scientists. And finally, modest investments in documentation and technology transfer can stimulate potentially impactful plant genomics research in Africa. And that brings me to the end of my talk. And I just want to mention that we have really received a lot of support. So I'm thankful to our collaborators at the University of Zimbabwe, Dr. Prince Maroa and Dr. Rose Masekesa. We also get a lot of support from the government of Zimbabwe, the University of Tuebingen, and also the Weigel Lab, in which we are hosted. And my team has done quite a lot of work towards some of the data that I've shown you today. But the most valuable players are these rural farmers from Zimbabwe. These are the farmers who very generously share with us their time, their seeds, and their knowledge. As you may know, in rural settings, you don't even make an appointment. You just turn up at the homestead. And so Prince has been doing this, and he has not been turned away from any homestead. Each and every time the people are willing to engage and to find out more about this project. So I'm really, really grateful for these farmers. Thank you.
Okay, so thank you very much, Professor Weigel and Dr. Patient, for the very nice introduction on the climate adaptation and the importance crop, which is the horn melon. So is there any question from the participants and students?
Okay, maybe before we have a question, I would like to appreciate Dr. Patience and Professor Detlef for the wonderful presentation. I hope this kind of presentation is going to help so much also our community and our students who are taking currently our course in plant synthetic biology. We have a course which we are running currently, and I hope some of the participants of that course are on this call. And the presentation will help them so much, and I would like them to ask a lot of questions from Dr. Patience, from Professor, to help them relate with whatever they have been learning and to help them improve or even on the future and on the projects which we hope to do with them. Thank you so much.
Yeah, very happy to ask questions, and you can ask me questions not just related to my presentation, but feel free to ask any type of questions that come to your mind.
Yeah, so maybe like while the participants are typing down or gathering their thoughts for the question, I have one question that I want to ask Professor Detlef briefly. So in our class, one thing that we want to do is we want to choose the candidate of the plants. So we are not trying to limit to just crops. We want to study plants that may be interesting in Africa. So we want the students to participate and identify what plants would be interesting. And this is including the feasibility of the plant genome sequencing that we are trying to study as well. One thing that I've been talking to Prachaya a lot is whether like we can learn about the biodiversity and also on the evolution of these plants if we're trying to sequence. Of course, if we sequence just one, it might be difficult, but if we sequence something that we know that they're morphologically similar to the others, that could be good. So that was a little bit like long of the intro, but like one thing I want to ask is that like we like from your presentation we see a lot of mutations that will be accumulated. I wonder like is it really, you know, like with those mutations, because some of the plants were multiploid, polyploid, I'm sorry. So were these mutations usually like go down to their offsprings, or a lot of them were just, you know, gone because they're just like mutated but not taken over to the offsprings or something like that. So I'm very, you know, not familiar with plants a lot. I work with microbes. So it's very different, I believe so. So if you can share further, that would be great.
Yes, I mean, sorry, but so I should have described that in more detail. So those were only germline mutations. So those were only mutations that were passed on to the next generation. We also are very interested in somatic mutations. So plants are really different from animals because as I'm sure everybody here knows, in plants, you can often regenerate an entire plant just from a piece of root or a piece of leaf or a piece of stem or whatnot. So somatic mutations can be meaningful in plants, and that's not just the kind of point mutations that I described, but also transposons. So that's a very long line of research. So Barbara McClintock, Nobel laureate 1983, I believe. She, was studying already somatic transposition in maize half a century ago, or longer than half a century ago. Yeah, so in terms of plants, so I think, and so maybe patients can also chime in there. I'm very impressed with the work that is being done with Solanaceae. So that's the family that to which tomato and potato belong, but also eggplant, for example. And so those species have a lot of advantages. The genomes are typically not too large. They are, they're larger than Arabidopsis, but it's not like corn, for example. The size is similar to rice or so, well, maybe twice as large as rice, but it's more manageable. Many of them can be transformed, and there is enormous diversity. And so some plants are highly domesticated and others are not so highly domesticated. And tomato itself is actually really super interesting insofar that it is really a major crop all over the world, but it has not been captured by major by multinationals like many other crops have been, which is also really interesting. What do you think, patients? So tomato and potato relatives, how relevant or interest, and eggplant relatives, how interesting or relevant is that?
I think it's super relevant, particularly for the tomato relatives, because you will find also in Africa quite a diversity of plants that belong to this group and are used for food as well. So yeah, I think it's really important.
I see. Yeah, I think tomato is also like a very sexy topic in the US right now as well, because like I think they just try to study more of it when they just chip the plant onto the ISS, the space station, because they're trying to the astronaut do something with that as well. I think that was like when I was talking with them at NASA. And the company that was started in the US, like Norfolk, has also established a new line, the germline edited of purple tomato. So I think like that was one of the example that we are looking forward to explore into in our class. where we are trying to think about if we do any engineering of the plant, can we like bring it back to the environment or bring it back to the community or not? So I think that that's a very important point. Kimberly is not here, but Kimberly, I believe, like she's originally from Zimbabwe. She's also the instructor for our class, and she is also working with tomato at Caltech right now. So maybe like we will talk more and we'll bring it together. Maybe we'll contact you back again to share some information further through email and see what whether like if there's any specific strains or specific types that we should look further into.
I think, yeah, so I think for plants that belong to Solanaceae, in my home country of Zimbabwe, we have quite a number of wild plants that are actually toxic, and I think this is an opportunity for genomic engineering as well. So they contain, nutrients that will be important to humans and animals, but they tend to be toxic, and therefore I think there's an opportunity there.
So Wamakuyo has a question in the chat, but before we get to that question, I wanted to ask Patience something, and really a question to everybody here in the call. So I just read about a project, or I've seen quite a few projects where people propose to domesticate new species, and so certainly what I have learned from Patience that there are a lot of species that have not fully benefited, if you can say that so, in that way, from the tools of modern breeding that are, half domesticated. So I sort of, when I read these proposals, I sort of question whether we should really, think about domesticating plants that have not been domesticated at all before, or whether it does not make more sense to focus on plants that are, on the verge of being fully domesticated. And the horned melon that Patience told us about is really a great example there. Patience, any thoughts on that? Domesticating new plants versus working with the ones that are already, interesting to people?
I'm all for domesticating the new plants. So
okay.
What we see with edible wild species is that very often they tend to become a food security fallback. So whenever there's a drought or famine for whatever reason, people are depending on these edible wild species for nutrients. So I think if they're already adapted, right? So why not get them into that value chain?
Okay, cool. Yeah. Wamakuyo had a question. So what measures are in place to track changes to climate change, and then to know about the original genetics of the plant? So this is really what you can do with herbaria. So that has become a cottage industry. So a lot of people are going to herbaria, extracting DNA from herbarium samples, and then looking at the genetics. And so the nice thing there is herbaria are really full of plants that have been sampled pretty much continuously over the last 200 years or so. So you can go back 20 years, you can go back 50 years, you can go back 100 years, and it's relatively straightforward to extract DNA from these herbarium samples. So they are really super valuable. And it's really a shame that when funds are scarce, that herbaria are often the first ones that are asked to make savings.
Okay, anyone with another question, please, if you have a question, unmute and ask, raise up your hand and ask, or type in the chat, and we shall be happy to have your question.
And also questions can be about the factors that could drive you guys into the point where you can see your education succeeded as well. Not only
so maybe I can ask a question. I would really like to hear from any of you. What would you say is the main thing that holds you back right now? And there are probably many different things, but I would just love to hear from you where you said, here is that one. If I had a magic wand, this is the one thing that I would like to change right away. For any of you, what would be the one thing that, with your magic wand, you would want to change?
Okay, starting with me, I would love to change the scientific research and innovation landscape across Africa, like how research is handled, how the policies are implemented in Africa, and how policies are debated, and how the government gets involved into the scientific world to improve it and to promote it, and with the final aim of promoting the African bioeconomy. That would really be something that I would be looking at for the near future, involving policymakers in improving the African bioeconomy and improving research and innovation.
I would agree with Kato. So I think we can make a great leap in scientific innovation in Africa if African governments put their money where their mouth is and pour more funds into scientific research and development.
Yeah, I think like others in the chat can chime in. But one thing I want to add into this discussion is the trust that we need between countries. So so far when we're trying to work on these kind of like let's say we are volunteers from outside of Africa, trying to work together with the African teams where Kato is like connecting with a lot of people. The first concern that come to a lot of our discussion is that like, is this really okay if we get in and work together with this sequencing project? Will these information be useful for the African people, for those who are in the area? And is it going to be another kind of like resource extraction from the Western, from the so to keep coming to the global south, take things away and not giving back to the community? So we're trying our best to not let that happen, right? Because we want to collaborate. We want to make sure that the resource is shared. We want to make sure the education is made accessible. So there's a lot of initiatives that we'll be touch on as part of a class. So we're gonna have like the final class on the international like relation, and also some other policy that will be helping on this angle. Like for example, IUCN has the digital sequence information, which is trying to make sure that the equity usage of these information, not just the sample, but also the origin of these data can be useful for everyone. So I think that's the part I want to raise and make sure that we can try to build more trust and make sure that if we are trying to go for the best use of the data as a scientific community, we should be trying to get into similar terms, not trying to just extract information and go for the base community we can make, something like that.
Yeah.
So there is one question in the chat box. So the second now. So is there a way your lab can collaborate on the various research in your lab with African scientists in the field? Have you and any of your colleagues in this agenda started this already?
Yeah. So I think this is mostly through supporting the research program that Patience is doing.
And another question, which is more about the research. How do you deal with the off-target effect in your gene editing approaches, like crops, like corn, melon?
So I would say that we are not conducting gene editing of horned melon. At the moment, we are not conducting a transformation of horned melon. Our approach is to study the genetic diversity by genome sequencing.
Maybe I can add to the question from Julia. So something that we have done quite a bit is also to host visitors who not only receive training, but then can also generate data. So for example, we had a visitor from Mauritius, which is a country which has quite good resources. And he was able to bring back to his country methods that we had developed for tracking herbicide resistance. So in Mauritius, very intensive agriculture and enormous amount of pesticide is being used. And he works for agricultural extension, basically. And so this had a direct impact by tracking herbicide resistance. And then we had another visitor recently from Burkina Faso who was also working, I think, patients that also would qualify as an orphan crop, Kirstein's groundnut, or is that a more established crop? Yeah.
Yeah.
So who came with a collection of Kirstein's groundnut, and we were able to produce a reference genome for her, and also generate information on genetic diversity. And then she was able to go back to her home institution, and she will now, use this information in her own research. So I really see what we do where. So I would say is less collaboration and more trying to catalyze to support research directly. So if that makes sense.
Okay, I also have a question, and it goes to Dr. Patience. I'm sorry, I have jumped in, yet there is another question from Ramadan and one in the chat. Dr. Patience, as SynBio4ALL Africa, currently our aim is basically to help African scientists and the passionate scientists to gain hands-on skills in whatever they are doing. So I would like you to maybe talk on this, like to a passionate African who is on this call currently, who is willing to gain more technical skills, more especially in the plant field. How can your project help on this beyond like what you're doing, but also help to build the skills? Because our aim is to see that Africans also gain their own skills, not only like shipping things to Europe, to US, but also trying to have the expertise locally present to do some of the things from Africa instead of always moving to other places. So how can your project help to tackle this, maybe starting from a small scale, like maybe in the southern part of Africa? Yeah, thank you.
Yeah, thank you for that question. That's a really important question. So I think that our project right now is very focused on one nation, and it is a bit difficult for us to train scientists from other parts of Africa because it's a very small project. What I would suggest for anybody who wants training in bioinformatics, in genomics, is that they should keep a lookout for the African Plant Genomics Network that is going to be coming online in the next few months. They can already follow the African Plant Genomics Symposium on LinkedIn. We will be having quite a few initiatives taking off the ground involving bioinformatics training, and we'll be having journal clubs and also focus groups on specific techniques, themes, topics that students across Africa might be interested in. And this training will be focused, these initiatives will be focused specifically on plant genomics in Africa. For those people who want genomics training that goes further than plant genomics, or if they are interested in animal genomics or microbial genomics or human health genomics, then I would direct them to the Africa Biogenome Project. The Africa Biogenome Project is doing a really good work of holding 3 workshops across Africa per year. So they target, no, actually it's 4 workshops. So they target 4 regions in Africa: North Africa, West Africa, East Africa, and South Africa. And they have 4 workshops in each region per year. And part of those workshops involves training on different aspects of genomics. So be it genome assembly, DNA extraction, library preparation, training on how to collect plants, what the standard operating procedures are for collecting plants, how to respect the bioethics around collection of plants and all of this. So within the context of our project, it's really very, very focused on Zimbabwe right now, but there are continental initiatives that these students can be directed to.
Okay, to add on that, we have some community members, like members of SynBio4ALL, who are based in Zimbabwe and some who are based in Malawi. Do you think like those ones based in Zimbabwe can touch base with you and see how best you can discuss with them about improving their skills in the field?
Yes, definitely. So for those who are based in Zimbabwe, and those who can travel to Zimbabwe at their own cost, we will have a training on library preparation and genome sequencing with Oxford Nanopore technology in this November. So they can get in touch with me and we can have a chat about how they can join that workshop.
Wow, that's very nice. Thank you so much for sharing this information. So I
Yes.
see one person raising the hand. So Dr. Ramahan. I'm sorry if I'm telling your name wrongly.
Yes, please. Good evening to you, sir. Yeah, my concern is about variability, phytochemical variability in plants. Phytochemical variability in plants, in a given plant. Whenever you plant, it is always not standard. So I'm normally what we have been doing when we are making phytochemicals, to standardize, we think of, for example, analytical markers. We identify a given phytocompound, and then we assume that all other phytochemicals are there basing on the other one. So I don't know how accurate that one can be. Then what would be another question would be, would there be any other method of standardizing, or maybe yes, standardizing or assuring the composition of the different phytochemicals in a plant, why owing to our methods, maybe biotechnological method that we can put in place to ensure that the plant can actually produce roughly the same amount of compounds every time we cultivate.
I think the only way is to exactly replicate the cultivating conditions. That's one of the annoying things about plant metabolites. They are super sensitive to both the life history and the culturing conditions. So that's what makes it so difficult to work with plant metabolites. If that's what you're doing and the variability is something that really impacts your research, I completely feel with you. So.
Should I assume that when we are doing analytical markers, like for example, I'm working on Phyllanthus amarus, I look at a phytochemical like phyllanthine and I standardize it. Then I assume that if phyllanthine is available in my extract in this amount, then other compounds are there. Would that one be really viable? Or maybe, and do these compounds occur in standard ratios, that when this is at this amount, the others are in the other amounts? So how can I guarantee every time I extract that yes, all the components are part basing on what I have standardized? Because I cannot standardize the entire, I pick one chemical, phytochemical to standardize against. So will I always assume that they occur in ratios so others are also there?
I'm afraid that I don't really have a really good answer there.
Thank you.
I think that question would probably best be answered by an expert in metabolomics.
Okay.
Oh,
Thank you.
okay. I think we have some people who are good at metabolomics we are in touch with. I think Ramadan, you can pass your question via SynBio4ALL@gmail.com, and we can push this question to experts because we have them in our network who can help us get the best answer. Maybe I have one other question to Professor Detlef. On this call, we have very many people who are still looking at building their careers. Like they are still trying to think about which career direction to take. They are not yet certain about the exact careers. In that path, what would be your talk about to a young African who is still thinking about a career path, but who has passion with plants and maybe such kind of field? What would you talk about that? What are the kinds of opportunities which are present globally to this young African who is still thinking about building a career, and how best can they utilize these opportunities, whether back in at the African continent level or at a level of the whole world?
So I obviously like, plant genetics. And one of the things that I think there's a huge opportunity, and maybe patients can also say something to this, is that much of the breeding that has been done has been breeding for optimal conditions. And I think to have breeding programs that are geared specifically towards conditions with limited inputs, I think there is huge opportunity. So that's not necessarily in the interest of big multinationals, but I think it would be very much in the interest of anybody who is thinking about a future world where we more responsibly use the resources that we have. Patience, any thoughts on that?
No, I think you put it succinctly. I'm not going to add anything to that.
Mm-hmm.
Okay, that's
so plant genetics is really it's an amazing area, and it's also an area where artificial intelligence already has been making a real difference already for well over a decade. So yeah, I think it's a fantastic direction to go into.
Okay.
And I personally, so one of the things why I think plant breeding is so important is that it is really for everyone. So food security is one of the most important things in the world for ensuring that countries are sustained and get along. So, and that's sort of my mantra always, that anybody living anywhere in the world should have as one of their main priorities to ensure food security anywhere else in the world, because this is really where it all starts. So what good is whatever medical treatment if you don't have food security?
Okay, thank you so much for that response. Maybe as we conclude, I just have also one last question. On this call, I'm sure we have some university lecturers because some of them normally join us on this call. What kind of, I wouldn't call it advice, but I can call it like add-ons. What kind of add-ons can you give to the university professors who are lecturing in different African universities who would love to improve the way they deliver their knowledge to the students? Because we know that most of the African universities currently, they are still teaching the traditional biotechnology. A few of them are handling the modern biotechnology simply because of maybe lack of equipment and maybe lack of resources to deliver the exact knowledge of the modern biotechnology. How would you try to maybe give some knowledge to those lecturers who are here? And also, in conclusion, how can you encourage these lecturers to improve the delivery system of their knowledge? And also, are there some global opportunities which these lecturers and also their students can look into to improve their knowledge, like maybe exchange programs, or like just of recent, there is an opportunity which I saw online and I posted it in the community of SynBio4ALL Africa, and it was from Max Planck, and one of our students has got that opportunity and is, I think, traveled today to Germany. So like such kind of opportunities, internship opportunities, maybe fellowships for lecturers to improve their knowledge and other students, all such kind of things which you could be having knowledge about. Thank you.
Yes, so I'm not sure it's my place to give, advice to teachers in Africa.
Not in form of advice.
But the advice that I would give is don't underestimate how many people all over the world are willing to help. And of course, many people you write to them, they will ignore you, or but there are enough people who will respond positively, and so that's one of the things that I've learned in the pandemic, how easy it is with these modern means of communication to connect with people all over the world. And one of the things that I would suggest really to you have to be you have to be able to take rejection in the form that people don't answer you, but don't underestimate there's still going to be enough people who will respond. So one thing that I would really suggest, write to people who you think do something interesting and ask them, are you willing to give an online seminar on your work? And I think you will find that it requires some engagement and so on, but I think you will find quite a few people who are willing to do this.
Okay, thank you so much, Prof, for catching up with us today. It has been really interesting, and I hope even the lecturers we have on the call today, they have learned a lot of things which they will maybe incorporate in their teaching systems, because I did my undergrad from Africa, and the biotech style we were studying, it was the traditional one. But when I came to Europe, I found that there is modern biotechnology skills which we don't learn back in Africa, but they are being taught here. And I hope we shall continue adjusting slowly. I will give back the microphone to maybe Ice or Big or anyone, because I think the questions are done and we can come to conclusion. Yeah.
So before I give it to Ice, I just would like to thank all of you for participating in this webinar. And of course, I would like to thank Professor Weigel for giving a very encouraging advice and courage to continue the initiative and the collaboration for the plant genetics and the plant genome sequencing study. And of course, I would like to thank Dr. Patient for giving us an insight, especially for the opportunity for the African students in the Plant Genomics Symposium. So thank you very much, and I would try to pass it to Ice.
Yeah, thanks a lot, everyone. Thanks a lot, Rashia, for leading this. Yeah, and very much appreciate both of you who take your time and be with us here today. I think everyone on the call is really appreciated on the knowledge you've been sharing. Also, those who cannot make it, they will be connecting through the recording that we will be sharing shortly. So one thing I want to emphasize is that Dr. Patience shared in the chat that if you want to stay connected, please follow African Plant Genomics Symposium on LinkedIn. I think that's something that, if you are interested in the plant genomics and you're in Africa, please make sure that you're within the loop. And finally, I think this is a great initiative for us to learn more from the team at Tuebingen. And I think we will have a lot of questions as we are trying to move further along with our genome sequencing initiative that we're trying to do from our team. So with that, I think it's a great time for us today, and without like spending too much time, and maybe like this is a good point for us to wrap it up. And hopefully we can see you again in the class next week, and also in upcoming webinars. We're gonna have like a few coming in.
And so maybe from my side, thank you so much for having me. And I think these sort of bottom-up initiatives like yours, we just need many more of those. That's one of the things that I've also learned at this symposium in Nairobi, to which patients invited me. That it's one should not underestimate the power of bottom-up initiatives. Why I of course agree that governments should do their part. They often do not, but don't underestimate what all of you can do.
Thanks a lot for the support. All right.
Okay, maybe Ice, before we conclude, I know for the people who will be watching this recording, please, if you have a question to either Professor Detlef or Dr. Patience, please send your questions via SynBio4ALL@gmail.com. You can check for the exact email on our website. We shall pass these questions to Professor Detlef and Dr. Patience, and you will get instant responses. Thank you so much, Dr. Patience and Prof, for joining us here.
Thank you.
so
Thank you,
much.
everyone.
Thank you very
Thanks a
much.
lot. Bye.
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 Bye. Let's keep in touch.